Welcome to the Explainer. Today, we're taking a deep dive into a bacterium that every medical microbiologist should know inside and out, Pantoea agglomerans. It's a fascinating organism, full of surprises and clinical challenges.
So, what is Pantoea agglomerans? Well, you can think of it as having a double life. On the one hand, it's this totally common bacterium you'd find just about anywhere in the environment, on plants and the soil. But on the other hand, it can be a really nasty opportunistic pathogen, causing anything from an infection from a splinter to a massive hospital-wide outbreak. It's this duality that makes it so clinically important.
Now, just a quick note before we jump in. All the information we're covering today comes from a comprehensive, deep search using Gemini, specifically tailored for the clinical community. If you want to explore the primary literature yourself, all the citations are right there for you in the accompanying notebook document.
Okay, first things first. Let's really unpack what this organism is, because its history, believe it or not, has a direct impact on the lab bench today. Understanding its taxonomy is key to avoiding some really common diagnostic pitfalls.
You know, this bug has had a bit of an identity crisis over the years. This whole taxonomic journey has been a real headache for clinicians and labs. You'll still find its old names, especially Enterobacter agglomerans, lurking in older literature and even in some lab information systems. And this isn't just trivia. It can seriously mess up surveillance data and even lead to misinterpreting an antibiograph.
So here's the bottom line for identification. Your standard 16S sequencing just isn't going to cut it for Pantoea. It simply can't reliably tell the difference between Pantoea agglomerans and its very close relatives. For definitive, rock-solid ID that you can trust for clinical work or an outbreak investigation, you absolutely need to use multi-locus sequence analysis, or MLSA. That's the gold standard.
All right, let's switch gears and talk about what this actually looks like in patients. It's really fascinating how the bacterium's environmental life gives rise to two very distinct types of human infections.
This slide really captures the opportunism of Pantoea agglomerans perfectly. Out in the community, you're usually looking at a traumatic inoculation. The classic example is getting pricked by a rose thorn, which then leads to septic arthritis. In that case, you've often got to find and remove that foreign body. But inside the hospital, it's a whole different ballgame. It's almost always an iatrogenic error, like a contaminated batch of IV fluids that causes these widespread point source outbreaks of bacteremia.
And you know, it's our most vulnerable patients who really bear the brunt of these healthcare-associated infections. We're talking about premature babies in the NICU who are at huge risk, and our immunocompromised patients, like those undergoing cancer treatment. For these populations, a Pantoea agglomerans infection can be absolutely devastating.
So you get a culture at the bench. How do you actually spot Pantoea agglomerans? Let's walk through the key lab characteristics, because while it can be tricky, there are definitely some telltale signs to look for.
Under the microscope, it's a pretty standard-looking gram-negative rod, but on the culture plate, things get interesting. On blood agar, about 3/4 of isolates will produce this really characteristic non-diffusible yellow pigment. It's a fantastic clue. But remember, that means 1/4 of them don't, so you can't hang your hat on it. And to make things even more fun, its lactose fermentation on McConkey agar is variable. It can be pink or colorless, so it can easily be mistaken for other bugs.
Okay, this is where the biochemical panel really becomes your best friend. Of course, it's oxidase negative like the rest of the Enterobacter, but the key differentiators are that it's negative for both indole and urease. That's how you're going to separate it from the usual suspects like E. coli, Klebsiella, and Proteus.
Now let's talk about resistance. And this number, wow, it's a big one. A staggering 84.8% of Pantoea agglomeranus isolates are intrinsically resistant to a very common workhorse antibiotic. And that resistance is to ampicillin. This isn't something it picks up, it's a built-in, predictable feature. It's all because of a chromosomally encoded AMP-C beta-lactamase. Now, this isn't like the inducible AMP-C you worry about with Enterobacter. This one is expressed all the time but at a low level. It's not potent enough to knock out your 3rd or 4th gen cephalosporins, but it absolutely makes ampicillin and amoxicillin useless. That is a critical piece of information for antimicrobial stewardship.
So, knowing about that intrinsic resistance, how do we actually treat these infections? The short answer is, you have to let antimicrobial susceptibility testing, AST, be your guide. There aren't any species-specific breakpoints for Pantoea, so we have to use the broader guidelines for Enterobacter.
And this is a super important directive right from EUCAST. When you're looking at an AST result for Pantoea agglomerans,
You need to apply the breakpoints for the entire Enterobacteriales category. This is how we keep our reporting standardized and, more importantly, clinically relevant.
Now, if we look at specific guidance, like the Dutch SWAB guidelines for sepsis, you can see a real emphasis on optimizing pharmacokinetics and pharmacodynamics. For the beta-lactams like piptazo and meropenem, they strongly recommend using prolonged or even continuous infusions. The whole idea is to maximize the time that the drug concentration stays above the bug's minimum inhibitory concentration, which we know can lead to better outcomes for patients.
For pediatric patients, especially neonates, it's a completely different story. Dosing is always weight-based, and the initial empiric therapy is usually much more aggressive, often using combination agents. For really severe infections, like meningitis, you'll see them ramp up the dosing frequency, like here with ceftriaxone, going from once a day to every 12 hours.
Finally, let's talk about prevention. And this is so important because in the hospital, Pantoea agglomerans is almost never spread from one person to another. It's almost exclusively a common source pathogen, which means everyone is getting it from the same contaminated item.
So, where is this thing hiding? Well, the main reservoirs are environmental and iatrogenic. We're talking about things that get put directly into patients, contaminated total parenteral nutrition, or TPN, IV fluids, even drugs like propofol. But you also have to look at the hospital's water system. Sinks, faucets, and especially stagnant water in the pipes can be major sources. This means our prevention strategy has to be laser-focused on things like sterile compounding and just really good environmental hygiene.
When an outbreak does happen, there is a tried-and-true playbook you follow. First, you identify a cluster of cases. Then, the epidemiologists get to work, looking for that common thread, a shared medication, a specific procedure. The next step is to culture those suspected sources. And then, this is crucial, you use molecular typing like pulsed field gel electrophoresis, or PFGE, to prove that the bug from the patient is the exact same strain as the one you found in the environment. Once you have that link, you can implement effective controls and stop the outbreak.
And this really brings us to the ultimate takeaway. Think of Pantoea agglomerans as a sentinel, an early warning system, because it comes from the environment. When we start to find acquired resistance genes in it, things like ESBLs or carbapenemases, that is a massive red flag. It's a signal from the ecosystem that these dangerous resistance genes are circulating out there and are making their way into the clinic.
So, I'd love to hear from all of you. Have you encountered Pantoea agglomerans in your lab? What kind of challenges has it presented? Please share your stories and your insights down in the comments. And if you found this explainer useful, make sure to subscribe for more deep dives into clinically relevant microbiology. Thanks so much for tuning in.